Hello everyone, welcome back to Dr. K. PREM-PRIMER lecture series presenting by Dr. K. PREM, that's me. In today's lecture, we'll talk about ampicillin resistance gene or beta-lactamase. As you all aware that this is the first antibiotic resistant determinant discovered, and this beta-lactamase provides the resistance to the bacteria from penicillins or ampicillins or beta-lactam ring containing antibiotics. So in today's lecture, we will talk about the various properties of beta-lactamase, such as what is the gene properties and protein properties, how this lactamase, beta-lactamase inactivates the penicillin, right? So these things we will study in today's lecture. The first thing I want to tell you in that is the beta-lactamase used as a genetic selection marker. So if you take any vector from the pBR322 to mammalian vector, the most popular and first choice antibiotic selection marker is beta-lactamase. Genetic selection marker helps in the selection of transformants from the non-transformants. Whenever you transform the bacteria with the certain plasmid, the plasmid containing bacteria will grow in the presence of antibiotic, and whereas the non-transformed bacteria will die. That's what is the duty of the genetics selection marker, right? So the beta-lactamase or ampicillin resistance gene is a part of many vectors. I'll give you a few examples here. One is the pBR322, which has the ampicillin resistance marker, and a pUC18 ampicillin resistance marker, and the PET21 ampicillin resistance gene, that's a beta-lactamase. So the genetic selection marker play a key role in the selection of transformants, from that selection of recombinants, right? So in addition to the genetic selection marker, origin of replication will be there. That's for independent replication of plasmid, and MCCS, multiple cloning sites, will be there. They are for they facilitate the cloning of foreign DNA fragments. So in today's lecture, we'll see the properties of beta-lactamase gene-wise and protein-wise, as well as the how it is going to hydrolyze the penicillin. So cloning vector, you see, pBR322 have a ampicillin resistance marker, which helps in the selection of transformants and followed by the recombinants. And pUC18 have a ampicillin resistance gene, right? And that's useful in the selection of transformants. And the PET21 also have ampicillin resistance gene. So here you see, this is what is the penicillin structure. And it is, the red one is a highly reactive beta-lactam ring, right? So this ring is involved in the inhibition of transpeptidase, then followed by the inhibition of cell wall synthesis. So here, the beta-lactamase is going to attack the beta-lactam ring and inactivate the penicillin, right? You see here, the beta-lactamase gene is ORF is having the 858 base pairs, and this also have a promoter, and the promoter is from promoter, and you see transcription initiation takes place at the -36 base pairs from the ATG, and there is a promoter. So the enzyme RNA polymerase will binds to the promoter and initiates the transcription, and the mRNA is produced. That mRNA converted to the protein upon translation, it will have a 286 amino acids. The beta-lactamase complete protein have a 286 amino acid. So when you look at the functional domains, it has two domains. One is a leader domain or signal peptide or beta-lactamase domain. So 1 to 23 amino acid are not found in the matured enzyme. So the leader domain, which helps in the transport of enzyme from cytoplasm to the periplasmic space, where it going to inactivate the penicillin. And the beta-lactamase domain is from 24 to 286 amino acids. And this domain is actively involved in the breaking down of beta-lactam ring of penicillin. So beta-lactamase have a active site. That active site is consist of serine amino acid. Serine is at the 70th amino acid is the serine. That's a part of the beta-lactamase domain active site. The beta-lactamase active as a monomers. Active as a monomers, it does not require any cofactor or a carbohydrate moiety for its functional status. As I told you that the beta-lactamase active site consists of serine, that's a 70th amino acid. That 70th amino acid serine with the OH, it breaks down the amide bond of beta-lactam ring. This bond is hydrolyzed. Once it is hydrolyzed, the penicillin is converted to the penicillinic acid. That's an inactive form of the inactive form. So it cannot inactivate the DD transpeptidase. So it will not mimic the as a D-alanine, which is the substrate of transpeptidase, which is required for the cross-linking of two polysaccharides of the cell wall. So here you see, whenever you transform the bacteria with the plasmid consists of a beta-lactamase gene, then if you grow that bacteria in the presence of penicillin, then the bacterial plasmid with the beta-lactamase can produce the beta-lactamase. That beta-lactamase can hydrolyze the amide bond of penicillin and converts into the penicillinic acid, which is an inactive form, and then the bacteria survives. This is how bacteria escapes the penicillin action from so that it can synthesize its own bacterial cell wall. Now you will see some other properties of beta-lactamase. Beta-lactamase catalyzes the amide bond of 4-membered beta-lactam ring. As I told you, that's a 4-membered beta-lactam ring, and render the antibiotic inactive against its original cellular target, cell wall transpeptidase. It's also called as a penicillin amido-beta-lactam hydrolase. It's also called as penicillin amido-beta-lactam hydrolase. It has 4 classes. It has beta-lactamases are divided into the 4 classes based on the active site. A, C, D classes are having the active site that the serine is the part of active site. And the fourth group is zinc-dependent or metallo-beta-lactamases. So this beta-lactamase, first it is observed or identified from the Salmonella paratyphi bacteria. It is having the one plasmid called as R7268. So this plasmid have the beta-lactamase, and in the plasmid there is a transposon called as Tn3 Transposon. In that Tn3 Transposon has the beta-lactamase gene. So then, as I told you that beta-lactamase has 3 promoters, 2 are anti-clockwise, 1 is clockwise. And the beta-lactamase from Salmonella paratyphi or which is and also clone into the pBR322, it belongs to the Tm1 beta-lactamase. So the Tm Tm1 beta-lactamase. And the Tm1 beta-lactamase, it belongs to the class A. And this promoter of the beta-lactamase is a constitutive promoter. The constitutive promoter means it expresses constitutively irrespective of the presence of substrate. If substrate is there or absent, doesn't matter. The same level of expression you can see in case of the beta-lactamase. And usually the class A beta-lactamases are having the average size of 25 to 30 kD. As I told you, they are active as a monomers. They don't require any kind of carbohydrate or cofactors. And they have leader peptide or signal peptide. And they have beta-lactamase activity is present in the 24 to 286 amino acids only. And active site is consists of serine, which breaks down the beta-lactam amide bond and converts the penicillin to the penicillic acid. And it provides the resistance towards the penicillins, cephalosporins, and other antibiotics with the beta-lactam ring. So this is beta-lactamase is a part of vectors where it is been using as a genetic selection marker. So this is all about beta-lactamase. So hope you liked my lecture. So if at all, try to subscribe my channel. That's Dr. K PREM-PRIMER for many updates. And if at all you like, try to give thumbs up. And if you have any doubts over the lecture, please write to me through the comment section. I'll get back to you once I see. Thank you all for listening. See you again with one more lecture. Until then, bye-bye.